Hi everyone, my name is Irene and I'm going to present the poster entitled Molecular and Functional Atlas of Sex Differences in Secondary Progressive Multiple Sclerosis, Neurons Characterization. Multiple sclerosis is a chronic, neurodegenerative, and autoimmune disease of the central nervous system, whose main hallmark is myelin damage. The patient's symptoms vary according to the affected neurons, and the disease is classified in subtypes depending on its temporal evolution. In detail, this work focuses on the secondary progressive subtype, where, as you can see in the first figure, firstly the disability fluctuates among periods of symptomatology and recovery, and then this pattern switches into a permanent affectation. In the subtype, as in the rest of them, we found sex differences in several clinical and epidemiological features. However, the molecular mechanisms underneath those differences are poorly understood. So to this end, this in silico research aims to describe molecular and functional sex differential patterns in secondary progressive multiple sclerosis, addressing the investigation of the neuron cell type, evaluating single nuclei transcriptomic data. This data was obtained by performing a systematic review in public databases and following the PRISMA guidelines. This second step was processing the selected data, which included several sub-steps that ended up with the cell type annotation. This step allowed us to identify the neurons we were looking for. Finally, we performed statistical analysis to identify sex differences in gene expression, functional profiling, analyzing biological processes from the gene ontology, and signaling pathways from the K database. As a result of the systematic review, we selected and downloaded one dataset with single nuclei transcriptomic data of postmortem human brains. There, we identify the main nervous tissue cell types, including neurons. These neurons were characterized, and the obtained results are the following. The first 5 genes most significant overexpressed in females compared to males are CAN-SL1, an enhancer regulator by acetylation, ARL17B, involved in protein trafficking, LINK-PINT, a long non-coding RNA, which plays a role in negative regulation of transcription, and both MAGE2 and SINE3 involved in synapses. On the other hand, males compared to females present overexpressed TMSB10, which is involved in the maintenance of the cytoskeleton, although A and GAPDH are implicated in the metabolism and previously associated with multiple sclerosis, and LINGO1 and NDRD4, which play a role in the resistance to neuronal cell death. Next, the functional profiling results unveiled that the top 5 most significant biological processes overrepresented in females compared to males were related to synapses, whereas the males had overrepresented terms related to protein transport and localization inside the neuron. Finally, the top 5 pathways with the higher number of effectors dysregulated were axon guidance, and the signaling pathways of RAS, guanosine and adenosine cyclic monophosphates, and AMPK molecules. This is relevant to highlight because here we see that each pathway we identified effectors up-activated in females and other effectors up-activated in males. This led us to disclose that even though the pathway is altered in both sexes, the protein effectors involved are different for each sex. With all of this research, the main conclusion I want to pinpoint is that describing potential sex-specific biomarkers in multiple sclerosis, as well as in other diseases, is key to approach them from a personalized medicine. Moreover, in this work, we unveil sex differences in multiple sclerosis neurons at each level of study. Further research should be accomplished to deeply describe their role in the disease. That's all, and thank you for listening.